Let's get started. Good morning, good afternoon, and good evening. Thank you all for being present for this LinkedIn Live organized by Netri. Just keep in mind that this live will be recorded and available in replay via Netri's platform. During the live, you can ask your question at any time directly on the chat, and do not hesitate to send us your question by mail also. I am Jessica Rontard, R&D scientist at Netri for more than 2 years now, and very happy to host these webinar sessions whose objective is to present our technological solutions to study the impact on electrical activity during the addition of a compound on the synaptic compartment using microfluidic technology. As you might know, Netri is a French private company based in Lyon. We design, manufacture, and sell standardized in vitro models using microfluidic technology, also called organ-on-chips, for neurological applications and others as cosmetic, for example. We believe that organs-on-chip are appropriate to reduce the cost and ethical burden of animal models while accelerating drug development. Such systems rely on both relevance of human cells and the ability to construct relevant and complex microfluidic architectures of the brain. This technology allowed us to have standardized and reproducible cell culture protocols adapted to the specificity of microfluidics. It is important to note that organ-on-chips offer the possibility to analyze the neural networks thanks to MEA placed below the Netri's devices in direct contact with cells. To do so, we are composed of a multidisciplinary team with several expertise. A fabrication team to produce custom microfluidic devices, a biology team to validate that the technology does not interfere with cell viability and functionality of cells, a digital team to offer added value and create specific software adapted to our device, and a team to support the need of our customers with a constant concern for the end user experience. At Netri, we offer, of course, microfluidics products, but also applications focused on neurological troubles, such as Alzheimer's disease and axonal regeneration. We also offer a R&D service and specific capabilities. Today, we will focus on one of the microfluidic devices' capabilities, allowing the isolation of soma and neurites thanks to microchannels to create an in vitro synaptic compartment. I will start with a brief introduction of the preliminary data obtained, and then we will continue with a direct testimonial of one of our partners. Neurons are composed of cell bodies, dendrites, and axons. They communicate with each other via nerve impulse that leads to the release of neurotransmitters that can be excitatory, like glutamate, or inhibitory, like GABA. The use of conventional in vitro techniques and in vivo models have allowed many advances in the understanding of the mechanism involved in synaptic transmission. As soon as we want to add compounds and study their impact on synaptic transmissions, microfluidics offers the advantage of compartmentalization of cell bodies, dendrites, and axons to create and isolate synaptic transmission compartments. In Netri, we have designed and manufactured an asymmetric microfluidic device composed of 3 compartments with a shift channel, allowing isolation of soma and neurite to create an in vitro synaptic communication. Indeed, the length of the macrochannel between channel 1 and 2 allows axon and dendrite to pass, whereas the length between channel 3 and 2 is axon-selective macrochannels. All Netri's devices are made with PDMS and have standardized design and fabrication steps, allows the generation of reproducible microfluidic device. The device is aligned to 1912 plate format and easy to use without pump equipment needed. The asymmetric device is compatible with confocal macroscopy, as you can see on this immunofluorescence picture, and also compatible with MEA recording for the neuronal network analysis. I will now present the preliminary data obtained in collaboration with French academic team. The objective was to develop an in vitro model to study the communication between immune cells and neurons by immunoassay and electrophysiological activity analysis. Concretely, our experimental strategy was to culture differentiated glutamatergic neurons from human-induced pluripotent stem cells in channels 1 and 3 for 20 days. At day 20, human dendritic cells have been added in one of the 3 channels according to the conditions. Here you can see illustrative pictures that highlight the characterization of human glutamatergic neurons in NET3 devices. For example, we have the presence of MAP2 positive cells, a marker of mature neurons, VGLUT1 for vesicular glutamate transporters, and the expression of pre- and post-synaptic markers. Note that all cell culture protocols are available upon request. As I mentioned before, the microfluidic device was combined with MEA to perform functional activity recording. We are able to demonstrate difference in the mean find rate, that means the average spike number during the recording period, depending on if the immune cells were added in channel 1, 2, or 3. This data underline that functional activity can be a relevant marker to study the immunoneuronal communication. Our data highlight an electrophysiological signal was transmitted between 2 compartments of glutamatergic neurons linked by synapse when neurons are exposed to primate dendritic cells. In parallel, we analyze the release of secreted factors in the supernatant. We note a higher expression of interleukin 6 in the channel in the presence of immune cells, also underlying a fluidic isolation between the channels. We also analyze the release of neuron-specific enolase, which is constant between conditions, showing the absence of neuronal damage. In addition, quantification of the expression level of VGLUT1 according to the conditions are in progress. All this work was done in collaboration with French academic team, in particular with Tristan Gabriel, a psychiatrist and PhD student. Tristan has prepared a video for us. Many thanks, Tristan, again for this video.
Hi everyone, my name is Tristan Gabriel. I'm currently a PhD student in the laboratory GMAP, G-A-M-A-P in Saint-Étienne in the medicine faculty. I'm working on mucosal immunity and link with the nerves in the big stuff of the microbiota gut-brain axis. My clinical field is on anorexia nervosa, and I'm looking for evidences in communication between immune cells and nerves. So I propose to work with NET3 and ask them to study the gut-brain axis and especially how to modelize in vitro the communication between immune cells and nerves, because in animal models it's really difficult to study this gut-brain axis because of its high complexity. Working with Jessica Ronta from NET3, they propose devices that allow to isolate some neurons, cell body in channel 1 and cell body in channel 3, and in the channel 2 the synapses. In this model we secondary seeds, MODC, so dendritic cell derived from monocytes, firstly primed with LPS from bacteria from our lab, And we study the signals, the transmission of the signals between the MODC to the nerves, and to assess how it can change the electrophysiology of the cell culture. For this, we use the MEA technology, and we find really interesting results of high potential of this culture to assess the transmission of the signals. We also use the supernatant to assay some biological molecules from the immune cells and from the neurons. Thanks to this, we can propose a poster session in the FENS 2022 in Paris with the collaboration of NETRI. And now, next step is to write a short communication about these results. Thanks to NETRI to invite me to talk about our works. It's really enjoying to work in NETRI.
We really thank Tristan and Stéphane Paul's team for this presentation. In conclusion, microfluidic technology allows to recreate a physiological neuronal network. Our technologies allows the combination of several readouts such as MEA, immunofluorescence, and immunoassay, despite the small volume in macrofluidic system. These neuro-organ-on-chip models could be used to test the effect of pharmacological compounds on immunoneuronal signal transductions implicated in a lot of pathology with neurological and behavior manifestation. The next webinar will take place next week, and we will present our webinar about our compatibility with electrophysiology to understand population influence, to target or stimulate specific cell type or connection, and to measure a network speed and directionality. Thank you very much for your time and attention. We have time for questions. Let's see if there is any question. As a reminder, you can post your question via the chat or send us an email for further contact.